This lecture on plant biotechnology is on the topic Agrobacterium mediated gene transfer. I am Sharuraj KM, Associate Professor, Government Science College, Hassan. Agrobacterium mediated gene transfer. So, before we get into the topic, modern advances made in the gene technology has made it possible to genetically manipulate the plants for crop improvements. This genetic manipulation can be done by introduction of foreign genes into plant cells. Today, a large number of plants are genetically modified to produce transgenic plants in many important agricultural and commercial crops. In nature, there are a number of organisms which have the natural ability to change the composition of the genome of their host organisms for their survival. One such organism that we are studying today is Agrobacterium tumefaciens, which changes the genome of the host organism after infection by integrating the DNA segment of its own into the host. So, this microorganism, Agrobacterium, is also known as nature's genetic engineer. Agrobacterium tumefaciens is an aerobic rod-shaped gram-negative bacterium discovered by Smith and Townsend in 1907. This causes crown gall disease of plants. This bacterium forms overproduction of infected tissues to form a tumor, and they enter the plant cells at the wound site, generally around the crown of roots below the soil surface. So, this is called as crown gall tumor. Agrobacterium tumefaciens contains an autoreplicative extrachromosomal DNA segment known as plasmid. The plasmid consists of genes that are transferred to the host plants, and this set of genes is referred to as tDNA. T stands for transferred DNA. So this tDNA gets transferred from the bacterial plasmid to plant cells genome. The plasmid is named as TI plasmid, tumor inducing plasmid. And this TI plasmid was first studied by Larenbeck and others in 1975. The tDNA expresses itself along with the normal host DNA. Tumor-inducing plasmid, TI plasmid, is a megaplasmid of about 200 kb and consists of tDNA, Vir region, ORI sites, etc. The T plasmid also has genes for the synthesis and catabolism of special type of nitrogenous compounds known as opines. Example, nopaline, octopine, manopine, etc. Other than that, it also has genes for auxins and cytokinins. Agrobacterium mediated transformation is a process of using agrobacterium to transfer a gene of interest into the plant cells, generating transgenic plants. Agrobacterium mediated gene transfer is also known as genetic transformation. This is a method used to introduce new genes in the DNA of plants. The TI plasmid present in agrobacterium has this tDNA, and Agrobacterium tumefaciens mediated genetic transformation requires the presence of two genetic components located on the bacterial TI plasmid. On the TI plasmid, there are two kinds of genetic material which are important. The first essential component is tDNA. This has about 25 base pairs of conserved material, genetic material, and it has imperfect repeats at the ends and these are called as the border sequences. Uh, the border sequences are referred to as the left border and the right border, as you see in the figure. The tDNA contains 8 or so genes that are expressed in the plant cell and are responsible for the overgrowth in the transformed cell. So this is the one which is responsible for causing crown gall. The second is the virulence region. The VIR region is composed of at least 7 major loci A, B, C, D, E, F, G that code for enzymes responsible for mediating transfer of tDNA into plant cell. The Vir gene produces nicks on the tDNA region at the LB, that is the left border, and the right border, and then the transfer DNA is sent into the plant cells. The Vir genes, which are involved in the transfer of tDNA, will not be transferred themselves. So these only help in the process of transfer. These Vir genes only mediate the tDNA transfer, and the transfer is regulated by the Vir gene products, especially the Vir A and Vir G proteins. These are the ones which are very important in the process of transfer. Now, when the TI plasmid is used for transgenic plants production, the genes responsible for oncogenesis, that is auxins, cytokinin production, and even opine synthesis, are deleted from the T region, and this is called disarming. So in these TI plasmids, the oncogenes located in the tDNA region are replaced by the transgenes. This is also referred to as exogenous DNA. By altering the TI plasmid's tDNA section and substituting desired genes between the repeats, the left and the right, the LB border regions, Agrobacterium can carry out genetic transformation. The transgenes can come from one plant species or from unrelated sources. The tDNA is integrated in the host plant's DNA, resulting in the transfer of the desired genes into the plant's genome. The transformed plant cells can then be selected and regenerated into whole plants using tissue culture techniques. Some examples of plant species that are genetically transformed by using Agrobacterium are alfalfa, asparagus, cotton, potato, soybean, tobacco, tomato, walnut, etc. So next, coming to the applications of Agrobacterium mediated transformation. Agrobacterium mediated gene transfer is a powerful tool for plant genetic engineering, as it allows for precise and targeted introduction of new genes into plant genomes. This technique has been used to develop crops with improved traits such as disease resistance, herbicide tolerance, and increased yield. The first important application is crop improvement. This is used to introduce traits into crops such as disease resistance, herbicide tolerance, improved yield. Then the next application would be functional genomics. This is used to study the function of genes by introducing gene constructs that alter their expression or function. Then the third application would be molecular farming. Agrobacterium mediated transformation is used to produce recombinant proteins in plants. Then the other application of this transformation process is biosynthesis of novel compounds. This is used for production of novel compounds in pharmaceutical industries, chemical industries, etc. These are some of the applications of agrobacterium mediated gene transfer. The references are given here. Thank you.